The central dogma of molecular biology states that DNA leads to RNA and then it leads to proteins. But what does that mean? Your cells, they carry chromosomes that are made of DNA, but the DNA only holds instructions for making a protein. That's all, it's just info storage. There is a process called protein synthesis in which the DNA is accessed for its instructions, like a recipe, and then the instructions are used to build a protein. So that explains the blue DNA on the picture on the right and the purple amino acid chain, but what's the pink? In this process, there's an intermediary that's used to carry the information from the nucleus in the DNA to the place in the cytoplasm where the protein will be assembled according to the instructions. All right, so I have Swedish lineage, and I have my great-great-grandmother's cookbook. It's in Swedish. I needed it translated, and doing it myself was a little slow going. So I found a woman on the internet that did translation services for Swedish to English. And unfortunately, she was on the West Coast, but I didn't really care where she was because I was not sending her my great-great-grandmother's cookbook. I made a copy, and I sent her the copy, and then she translated it, and then she sent me back the English version. And I then made wonderful Swedish recipes like curried egg salad and salted cod. Yeah, great. But the point is that I retained the original, like the DNA that stays in your nucleus, and I sent out a copy to be translated into this new language, just as RNA, or that pink on the diagram, carries the instructions for making the protein to be translated from nucleic acid to amino acids. What does this mean for you? Your DNA holds the instructions for the proteins that make you, you. For example, there are pigmentation genes in your chromosomes. They carry instructions for making a pigment of a certain color. And then that pigment can be made and inserted into your eyes to be expressed as your eye color for the world to see. Taking the instructions in DNA and making it into RNA and then into an amino acid sequence, it's a two-part process. First, we have to make a disposable copy of the DNA that can travel out of the nucleus and then get destroyed, and we don't care. This is like me making that copy of the cookbook, and this process is called transcription. The word transcription just means to copy. This is what you do when you copy somebody's notes if you miss class. You transcribe their notes. More specifically, we're going to make something called mRNA, with the M for messenger. This mRNA is a disposable copy that is produced in transcription. And this is what is taking place in the top part of this picture in the nucleus of the cell. This is a picture of transcription. That big blob there it's not the nucleus, although we're going to assume that the process that you see here is taking place in the nucleus. The big blob is an enzyme called RNA polymerase. And just like DNA polymerase, RNA polymerase makes the RNA polymer. It knows the base pairing rules too. But RNA polymerase thinks that adenine will always connect with uracil, whereas DNA polymerase thinks that adenine and thymine always pair up. What's happening in this picture is that the RNA polymerase has broken the hydrogen bonds in the DNA, and the DNA has unwound in this little spot. Here's one DNA, here's the other strand. The DNA has unwound in this little spot where we know that there's a gene we want to make a protein from. When we unwind the DNA, we have two strands of DNA, but we aren't going to take the sequence from both of them, just from one of them. The strand holding the gene sequence is called the template or coding strand. And this is the strand that will hold the gene that we want to make a copy of. The top strand here that I left in yellow is called the non-coding or non-template strand, and it just kind of hangs out. When we think about mRNA, we have to think of it as sets of three. You can't think of mRNA as one long list of GTA, GGC, but you have to think of it as a short list of three nucleotides. Three nucleotides on mRNA are called a codon. I want you to write this down, and I want you to burn it into your brain. I don't care, write it on your hand. Codons are on mRNA. The mRNA will then leave the nucleus and travel in the cytoplasm until it reaches an organelle called a ribosome. Here in the ribosome, this is where we're going to take the sequence of nucleotides and translate it into a sequence of amino acids. To do this, you need a decoder ring. I, for my cookbook, was using a Swedish to English dictionary and decided that I needed a translator. I needed someone who knew both Swedish and English. And here in the ribosome, we need a molecule that knows both nucleotide language and amino acid language. This that we're describing here in general is the second part of this process of making a protein, and this is called translation. The actual decoder ring is little molecules called tRNA or transfer RNA, and these are the molecules that can recognize a sequence of nucleotides and say, oh yeah, this is the amino acid that corresponds. Before we get into the process, let's meet the translator. tRNA is the molecule that can both interact with a nucleic acid and with an amino acid. The picture here on the left is actually a pretty realistic picture of tRNA. It is a strand, it's just a strand of RNA. It's just a sequence of nucleotides. And it has a really messy shape, but it has two places on its shape that are very important. On the tRNA, there is a place called the anticodon. This anticodon has a sequence of three nucleotides, and let's say that the sequence is UAC. And that sequence will obviously match up with some kind of codon on the mRNA at some point. Remember, codons are on the mRNA, anticodons are on the tRNA. Interesting. Also, the tRNA has a site on it that will match up with an amino acid. Which of the 20 amino acids? Well, that's predetermined. But each tRNA has a specific anticodon and a specific amino acid. Think of it this way. If you're a tRNA molecule, you have an anticodon and you can only have one amino acid that goes with it. It's like a lock and a key. You don't multitask. For example, a tRNA that has an anticodon that is UAC will always carry an amino acid called methionine. I taught from a book a long time ago that has a picture of tRNA on the right here. I kind of like this picture. By using this cylinder shape, we can clearly see that at the top of the cylinder we connect with an amino acid, and on the bottom of the cylinder we have an anticodon. Let's look at the process of translation. So this blue blob here is a ribosome, and ribosomes have a large subunit and they have a small subunit, and you can see that here in blue and brown. They actually float around in the cytoplasm separately, and they only come together when they meet or dock an mRNA. So it seems in this picture that we have a strand of mRNA that is sandwiched between the large and the small subunit. Yes. And now the tRNAs are going to interact or read the mRNA and bring over the correct amino acid to the ribosome so we can make the right protein. Let's start. We have the first codon on our mRNA, and that first codon seems to be AUG. That's actually the first codon for every protein. So the ribosome reads this codon and it kind of calls out like The Price is Right, like I got AUG. Does anybody match with AUG? And somewhere out in the cytoplasm there's a little tRNA that has the anticodon of UAC. And that tRNA says, oh, me, I match with that. I can fit my anticodon with that codon. And so the tRNA comes over to the ribosome, and what he does is kind of drops off his amino acid and gets the protein chain started. All right, so now let's read the second codon. And we call for the matching anticodon. And we have the right tRNA bring over the right amino acid. So we can do this as many times as needed to make a growing amino acid chain, which is the first level of protein structure. In looking at the process of translation, it occurs to students that there seems to be a very specific match from codon on the mRNA to anticodon on the tRNA, and then to amino acids on the protein. Yes, in fact, this relationship is set in stone and reflected in this very strange grid here. This grid, this is the genetic code. It's the decoder ring. It's the Swedish to English dictionary. It is a list of codons on mRNA and the amino acid to which they always match. For example, the first codon in our example that we did on the previous slide, we had AUG, which ultimately brought an amino acid called methionine to our protein chain. How did I know that it was methionine? Well, I mean, it's highlighted in green on the grid, but also I noted that there are labels on the sides and top of this grid. So looking along the left side, I find the row with all of the codons that start with A, our first letter. And now I look along the top of the grid and find the column with all of the codons that have U, or our second letter. And then I find that row and column intersection, and I can look at a short list of 4 codons to find the one I'm looking for, AUG or methionine. The grid here, this is the decoder ring. This is the dictionary of Swedish to English that I needed to translate my cookbook. So what's kind of interesting about this is that if you can see, even though one codon will only have one amino acid to match it, interestingly enough, one amino acid can match to a few codons. And this is what allows the flexibility to create an unlimited number of proteins for life, even though we only have 5 different nucleotides and 20 amino acids. All right, so let's think about mutations for a minute. A mutation is something that affects the original DNA sequence. And this can be acquired in meiosis or mitosis, or just plain old mistakes in making the copy. Mutations don't have to be bad, but let's discuss one that does lead to disease. Every human has a gene that says make a red blood cell. Actually, it's a bunch of genes, but to keep things simple, let's just say it's one gene that determines if your red blood cells are nice and plump, like the one in the top picture, and are able to carry oxygen, or if your little red blood cells are sickle-shaped and not so great at carrying oxygen. There is a variant of the gene in which the nucleotides have been changed. One nucleotide changed. Somewhere in the sequence of the DNA in this gene, a thymine got replaced by an adenine. but that changes the codon. And then we would need a different anticodon to match it, which would then change the amino acid. In people with sickle cell anemia, there is one amino acid out of thousands. And that amino acid change is the genesis of their disease. But what if the nucleotide changed and it didn't lead to a different amino acid? Because remember, one amino acid had many codons. Actually, that's fine, that's great. That's what's called a silent mutation. And although your DNA sequence has changed, it doesn't really affect the processes in your body.